Everyone. Welcome to Barcode Biotechnology and welcome to the session of Drug Design and Drug Discovery Program. So basically, in this session, we are going to study about what is drug design, what it means by drug discovery, and also we will go to learn about why this is the booming section or booming career section in nowadays. Okay, so let's start from the begin, that is from the introduction. So basically, as we have seen here, we have two main terms, that is drug design, and second one is the drug discovery. What is the difference? What do you mean by drug design? So design means preparation of any sort of compound, preparation of any sort of compound, and drug discovery. So to prepare this compound, first we have to find out the target. Okay, so let me tell you the introduction. So drug discovery and drug design focus on identifying the biological target and developing molecules that can precisely and safely interact with them. Here the them means the target. Let's up I am giving you an example here. Let's take an example of flu. Okay, flu. Flu is caused by the rhinovirus. So here the rhinovirus is our target. Okay, rhinovirus is the target. Cure To cure the flu or to prevent the rhinovirus, what medicine is given in the market? That is the paracetamol. The drug that is given, that is the paracetamol. But this paracetamol is having a drawback. After taking more than 7 days, it is find out that it may can leads to the hepatotoxicity, mild hepatotoxicity. So basically, what is hepatotoxicity? Hepatotoxicity is nothing but the liver problem. Okay, so in case of flu, to prevent the rhinovirus which cause the flu, we give the medicine that is called as a paracetamol. So what here comes the drug discovery or the drug design. So we have to prepare such a compound or we have to prepare such a medicine which is having the same effect, and that is the paracetamol, or it will have the more effect than the paracetamol with having no such side effect like liver toxicity or hepatotoxicity. So this is the drug discovery. This is the drug design. We have to prepare a moiety or medicine. Every medicine have their own structure. The paracetamol is having a structure, and we have to prepare one medicine that is having a structure or it is having a safety interaction with the receptor. So this is the introduction about drug discovery and drug design. First we discover a moiety, then we prepare or design the compound. Okay, so next point, that is a computer aided drug design have been become powerful tool that accelerate identification, enhance lead optimization, and increase the overall success rate in pharmaceutical research. So basically what it means, we cannot go for the synthesis of each and every moiety. We just perform the in silico study, which we will be studying later. What is in silico study? Or we perform some sort of software. We use some sort of softwares. By using some sort of softwares, we predict that whether the drug molecule which we have hypothetically designed will be affecting the receptor. Okay. So in this session, we'll explore how these integrated strategies enable faster and more efficient development. Okay, taking to you in the next slide. So here comes the main point. Listen carefully. If you have pen and paper, write it down because it will be very much helpful for you when revising or when you could just go through that. Okay. So in the drug design process or the CADD, computer aided drug design, we have five major steps. So the first step is the preparation of the ligand molecules and preparation of the target molecules. In the second step, we have to identify the binding site, not the target site. we have to find out the binding site. Then we perform the in silico analysis, which is also known as the molecular docking. Then we will find out the results. We perform the docking, find out the result, then we evaluate and validate with the marketed compounds. So basically, this is the point that first we have to prepare our ligand molecule. First, we have to find out the target molecule, then go for the binding site. If suppose, okay, suppose the let's take an example of many e very easy example, that is DNA gyrase. It is an enzyme which is responsible for bacterial infection. Okay, bacterial infection, DNA gyrase. Okay, so what is DNA gyrase used for? Okay, so how we can prevent the DNA gyrase? So we have a drug that is called as the anti-tuberculosis drug rifampin. We have rifampin. We have antibiotics, that is amoxicillin. These two drugs prevent the DNA gyrase. Okay, so here the amoxicillin is a ligand molecule and the DNA gyrase is the target. Okay, so in CAD, when we perform the molecular docking and we find out a result, okay, so in this process the target and ligand molecule interact and we get a result, then we evaluate that results and validate the result. So now the question comes, how to prepare the ligand, how to prepare the target, then how to perform the molecular docking. All your questions will have the answer in the next slides. Next, so what are the tools that are used for CAD? So what are the various tools that we are going to use and get the results? Okay, so first tool we have, these are the set of tools that we are using for computer aided drug design. So very first tool, that is the this one, that is the chem sketch. This is basically used for the preparation of the ligand molecule. Preparation of ligand molecule. So to prepare the ligand molecule, we have two tools. This is the chem sketch, and this is the chem draw. So these two tools are freeware versions, and this is used for the preparation of ligand molecule. Then for preparation of the preparation of target, we have two tools. One is Autodoc and another one is our Chimera. So this is the Chimera and this is the Autodoc. So these two tools are used for the preparation of target as well as to perform the molecular docking also. Now these two tools, Autodoc and Chimera. So basically what's the role? To prepare the target and to prepare the molecular docking, perform the molecular docking. Next, we have another software for the molecular docking, that is, it is called as the Pyrex. So, okay, this software is also for the molecular docking and for the 3D visualization. We have discovered what is 3D visualization. We will go further and we will discuss for the 3D visualization, the 2D interaction, 3D interaction, hydrogen bonding, all these things we are going to study in Discovery Studio. Okay, so all these are the tools which are used for the CAD. Apart from this, there are another softwares which you can also go through, that is the Swiss ADME. This is another tools. Then we have Swiss target predictor, Swiss target predictor. Then we also have tools like Gaussian. It is not used for the molecular docking, but you can go through. Then we have cast p. Okay, so these are some of the tools you can go through. Also there is, for the toxicity prediction, we have some tools like called protox. Okay, so coming to the next slide, The first question, identification and validation of target. So basically, how to identify any target. See, now in our society, many diseases are there and many works are going on that such targets. We have, you all guys know that about ICMR, Indian Council of Medical Research. We have our CSIR labs. We have BRIC, where it's all these three labs are working continuously on different type of target. Some lab work on malaria, some labs work on herpes, some cancer. These are the central government organization, and there are many works on leishmaniasis, many works on leprosy, some are under integrated medicinal technologies. Okay, so we can see various targets. Target, they may be simple like flu, from flu to epilepsy, anxiety. There are various targets. Target means nothing but the disease. What is the disease? So this is the target. So how to identify them? How to validate them? Okay. So for that, before going to target, let me teach you some key technical terminologies that are used in the drug design and drug discovery. Okay. So first, that is the target. I have already told you what is target. The target may be some simple flu to a cancer. Okay. So these are the targets. The disease, the underlying disease, and inside the disease, what is the receptor responsible for the disease. Okay. Then the binding site. Here you can see, let me show you this. This is the receptor. Okay, this entire thing, it is one of the receptor. Okay, so if you can see here, there is a ligand molecule is attached here. This one, the one I have circled in yellow. So this is called as the binding site, the point where the ligand molecule binds to the target. It's the point where the ligand molecule is bind to the target. So that is called as the binding site. Okay, here it is also written that the region on the target where the ligand interact. Okay, so what is pocket or cavity? It is also known as the Q site. So basically, Q site is nothing but this is a pocket. If we take an example of enzyme, let's suppose this is an enzyme. We have to formation of the enzyme complex. So as you know, that is the lock and key theory. So let's suppose it is the lock and this is the key. So this will be the interactive position and this is the pocket where the key is gonna bind. So it is a 3D hollow region. Okay, remember the point, 3D. Okay, so it is a 3D hollow region where the ligand molecule fits, that is called as the pocket or cavity. Then we have another term, that is the allosteric site. It is a non-active site where ligand binds to modulate the function. Okay, so basically what is it? Allosteric site is where the ligand molecule binds and shows the activity. So basically we have two type of activity. One is agonistic activity, another one is the antagonist activity. So what is agonist activity? So basically when the drug molecule comes and binds to the receptor, there is some changes in the receptor taken place. So taking the example of flu, when you take paracetamol, it goes and binds to the receptor of rhinovirus. There is certain change in the receptor takes place in the rhinovirus. It shows, if it shows agonistic activity, means if it curing your disease, if it is decrease your flu level. Okay, that is the agonist activity. It is a pharmacological term basically. and there is an antagonist activity. If you have taken the medicine, but what happened? The flu increases, and instead of having less cough, you are having more coughing. You are having more temperature in your body. That is the antagonist activity. Antagonist is nothing but the opposite effect. Okay. Then we have the next point, that is the drugability. Drugability means the target can bind drug molecule, likelihood that a target can bind to the drug molecule. Okay, we will discuss it later in brief for this drugability. And the pharmacophore is the 3D arrangement required for ligand binding. So if you can see here, let me draw a structure of benzene containing the CS3. This is called as a toluene. So you can see this is a 2D structure of toluene. If I show you in a 3D format here, so benzene ring containing a like this bond is called the dash bond, means it is present behind, not forward, and this is called as a wedge. Okay, in dash it is present backward. Okay, this means this side. So there is a 3D arrangement of this toluene is there. So for to be a pharmacophore, the ligand molecule must have the 3D arrangement features like hydrogen bond donor. It may contain hydrogen bond donor groups, acceptor groups. It may be hydrophobic or hydrophilic, aromatic rings. These are the things that is required for to be a pharmacophore. Okay. Next thing, now let's study about ligand molecule. We have studied about target, what is the pocket, what is Q site, allosteric sites. Now talking about the ligand molecule. So ligand molecule is nothing but it is from very small, like phenol. Phenol is such an antiseptic. As you know, that is an antiseptic. From phenol to such type of ligand molecules. This such big chemical structure. It is also a ligand molecule. Okay. So ligand molecules from the small molecules or large molecules that binds to target. So that is called as the ligand. Scaffold, the core structural group that is present in the molecule. Here you can see this is the structure which is I have given previously example. This structure is the amoxicillin. Okay, this compound is amoxicillin. So here in amoxicillin, it is a beta lactam derivative. It is beta lactam derivative. So here the main scaffold is this one. Let me show you in red. You can see that this one, this moiety, this is the beta lactam ring. Okay, so this is basically the beta lactam ring. We have many other scaffolds are also there. Let's suppose we have here, this is the pyridine. It is a scaffold. We have, then we have this structure. These are some heterocyclic structures. It is, let's suppose, furan. Furan is there. Pyrimidine is there. Pyrimidine. Many pyrimidine derivatives are used for the treatment of cancer. Okay, you know 5-fluorouracil. It is an anti-cancer drug. We have pyridine is there. Then we have pyrazine is there here. Okay, so these are some of the scaffolds. There are many scaffold, many. Then we have benzthiazole is there also. So these are the called as the scaffold. Scaffold is the core structure of the molecule or the core heterocyclic moiety that is present in the whole compound. Then the functional groups. It is the most important one. What is functional groups? Functional group, it may be nitro, it may be amines, it may be ether, it may be ester. So there are various functional groups are there, carboxylic acids are there, then alcohols are there. So the functional groups are helpful for the activity of ligand molecule. Let me give you an example as I have given you here an example of phenol. Let me clear this slide first. Okay, now it's clear. give you an example. Okay, so let's draw one structure. Pyrimidine moiety. We have the pyrimidine ring containing the OH group here. Okay, so if we perform this drug's docking, sorry, this is the pyrazine moiety. I have wrongly done the structure. Okay, so let's suppose this moiety having OH group here. It will show some docking result. Okay, and I change this functional group. Okay, I change this functional group from OH to CS3. It will give another docking result. So basically, what is the functional group role? That these functional groups helpful influencing the reactivity and binding. They change the binding position. They may change the activity. They may change the drug effectiveness. Okay. So if this compound is showing minus 5.0 result, the docking score always remain in the minus. I will show you that. Let's suppose this shows minus 5.0. It may show minus 7.5. That means this drug is having the more effectiveness compared to this one, right? So this is the functional groups do. Okay. Next, the smiles. Smiles is nothing but it is not like this smile. This one. This is not like this. It is a linear text representation of the chemical structures. Means, as you know that there is an IPSC name, there is a molecular formula. Just like molecular formula, we have to represent all the structures with a smiles. Means, instead of drawing this pyridine moiety, I can simply just write in a binary like representation C, then there is another C1. I will show you how to write it. We generally do this by using the Swiss AdMe. But there is another software which is internationally used, that is the PubChem. Okay, it is a chemistry database where you can find out all the smiles, all the marketed, all the reported drugs by the researchers. Okay, by just simply writing. If I write the amoxicillin, it will show you the amoxicillin structure. It will show you the amoxicillin smiles. Okay, then next is a library. We have next, it is the library. So library is nothing but collection of molecule for the screening. For the molecular docking, we have a set of library, means a set of 20 various compounds. Okay, that is called as the library. Let me show you here to clear the slides. Let's talk about library. See, we have one compound. Here it is the phenol ring containing the nitro. Now I replace this phenol nitro group into NH2 group. Then third, I replace this NH2 group with no substituent, no derivative. Again, here I have replaced the nitro group with the Cl, chlorine. Okay. By using this sort of things, we have prepared how many libraries? We have for 1, 2, 3, 4. For representation, I have just given you an example of 4 derivatives. These 4 derivatives are 4 set of library. It is a set of 4 compound. Okay. So library is nothing but a derivatives. It is just a derivative of the scaffold. Okay. Here the phenol is a scaffold and derivatives like chlorine is a substituent here, nitro is the functional group, NH2 is the functional group. Okay, so I can prepare from 20 to 100 set of libraries for the molecular docking. Next term, that is a virtual screening. It is a computational method to screen millions of compounds. Okay, first thing we know about what is ligand molecule. It may be from a small phenol to large derivatives. Okay. Then we know about the scaffold. Scaffold is nothing but the moiety, major moiety present inside the ligand molecule. Then we know about the functional groups. So how functional group affect the activity. Then we study about the library. Okay, we studied about library. So library can be prepared by using different derivatives of the scaffold. Then virtual screening is nothing. It is just the representation or the molecular docking of the library which we have prepared. So now the final key terminology is docking. Docking basically the interaction of receptor and the ligand molecules. Okay. Then the pose. Pose means the predicted arrangement of the ligand inside the binding pocket. So here if we have the pocket, if the ligand molecule have been placed like this or it placed like this, like here, let me show you bonds OH. Whether it's placed like any other form or it is not inside the binding pocket. Okay, that may be possible. If the drug is not having any such activity, it will not present in the binding pocket. Then the binding affinity, which I have told you earlier, that the binding affinity is the interaction, stronger interaction between the amount of interaction of receptor and the ligand molecule. It may be from -1 to -15. Okay, that is the binding affinity. How strong the bond formation takes place between the receptors and the ligand molecule. Then the grid box. So grid box, we have to select a particular region in the receptor. As I have shown you, you can see here that this is a GIF. Let's suppose I have prepared a grid box for this area only. So in this area only, how the ligand molecule interact with the receptor. Okay. So I will give a setup area then of the receptor and I will perform the docking, and inside this how the ligand molecule is binding to the receptor, that is the binding affinity, and this is the grid box, and inside how the drug is posing. Okay, then we have two types of docking are there. One is rigid docking and the flexible docking. In rigid docking, shortly just I am describing what is rigid docking. Rigid docking, that is we have protein fixed, ligand molecule flexible, means we have our protein just like let's suppose dihydrofolate reductase. It is a bacterial protein, and we just uh, offer a library set of library, means 5 to 10 derivatives. We just perform docking. That is called as a rigid docking. In flexible docking, what we have to do, both the ligand and link a protein is having flexibility, means we can change the sequence of amino acid. Change the sequence of amino acid. Basically, we have 21 amino acids in our human body. Let's suppose I just change the glycine. Okay. Where is my cursor? Okay. So in the receptor, let's suppose I have glycine at 10th position. I change the glycine 10th into leucine. Okay. So it leads to flexibility inside the protein. Okay. And we just perform the molecular docking using this leucine and using this reported glycine. And we compare the result. If mutation occur, mutation is nothing but change in the behavioral or change in something gene, change in the DNA sequence. So basically, this is called as the flexible docking. Now, what is drug discovery? We have already learned, and we also have learned about what is the basic key terminologies inside the molecular docking. So drug discovery increase productivity, new So increased productivity, drug discovery is scientific process of identifying new medicines. So basically it is nothing but identification of new medicines that can safely and effectively treat out the human diseases. So to treat the human disease, we need some sort of medications, we need some sort of new moieties, new medicines. So for this, we need the drug discovery. It is for it involves what it involves the disease biology. First, before going for the drug discovery, we have to find out the receptor and we have to study the pharmacology, how it interacts, what are the marketed medicines are there, and then how which portion of that organism or the receptor is responsible for the disease. Then identifying the target. First, we find out the organism. Okay, we find out the organism. Then we go for the target receptor. Okay, we go for the target receptor. Then we prepare molecule according to it. Designing or discovery of active drug-like compound. Then we, after preparation of the ligand molecule, we go for the efficacy safety interaction. We perform the docking, we validate the result, validation, we validate the result, then we go for the interaction, 3D interaction, 2D interaction, hydrogen bond donor activity, hydrophobicity, is it crossing to the blood-brain barrier? All these things we describe, then we go for the clinical trials. Then what are the other points are there? Seamless integration means just increasing the user friendly, to make it user friendly, enhancing the user's experience, scalable for the future growth. We have to see if there any future growth is there, user friendly learning. So now just like this platform where you learning about the molecular talking. Okay, so for that drug discovery is needed. Okay, so new thing come to our mind and new thing we are learning. So basically this is the drug discovery. So why the drug discovery is needed? Rising drug resistance. Rising drug resistance antibiotics. You can see in India, 10 out of 100 people is having the antibiotic resistance. Okay. See, if someone is having the antibiotic resistance and he is having cold, flu, all these type of diseases, in that case we have to give them antibiotics. The most common antibiotics which is provided nowadays, it is the amoxiclav. It is a composition of amoxicillin plus clavulanic acid. Okay, so if this is having the resistance, then how can we treat a person? How can we treat that disease? How can we cure that disease? So for that drug discovery is needed. We have find out that this is the receptor, this is the target, but we don't know what is the alternative medication for that. So there comes the drug discovery. There we have to prepare, we have to go through some sort of research, find out the exact molecular moiety that is needed. Okay. Next, emerging diseases. Okay, emerging diseases. Back in 2020, we have COVID-19. It is it just changed the world. It is the pandemic that changed the revolution. It makes a revolution in the field of drug discovery, in the field of pharmacy research. Okay, so COVID-19 breaks all these things, and there is booming of this drug discovery, drug research, bioinformatics. All these things are booming right now. COVID-19 changed all these things. There is some sort of disease called as black fungus. Okay, it is a fungal infection, or also it is known as the mucormycosis. It is called as the mucormycosis. Okay, unmet clinical need. The cancer, for cancer we need some medications. Alzheimer disease, it is a CNS related disease. Okay, scrambling of slurry speech, scrambling of the hand, unable to move the feet. So these are some sort of things. Alzheimer, they forgot what they told. Okay, loss of memory. So these are some of the diseases, CNS related disease and cancer, COVID-19, black fungus. We have leishmaniasis and final need of safe and affordable, more skill selective drugs. Okay, safe drug we need, affordable. How to make the drug affordable? We just prepare a molecule that is very much affordable, very much easy to synthesize. Okay, so these are the points that are needed for the drug discovery. Now coming to key steps in drug discovery. So what are the various key steps that are for used for the drug discovery? First thing, identification of target. Yes, we have to identify the target. Then validation, lead discovery, lead optimization, preclinical, clinical trials, and finally approval. Okay, this approval is given by the FDA, Food and Drug Agency, that is United States. Okay, so agency, that is the FDA, that is for also known as the US FDA, United States Food and Drug Agency. Okay, so let us go one by one about what one by one. First one is the target identification. Target identification. As I have given you the example, let's take an example of dihydrofolate reductase. Okay, dihydrofolate reductase. So it is a enzyme. Bacterial enzyme. So we have to first we identify that dihydrofolate reductase. It is a bacterial enzyme. Okay. Now after the target identification, the next process is target validation. We identify the bacterial enzyme, then we go for the validation. For validation, what we have to do? We just have to, so for validation, we just need to just find out the what are the marketed drug available. Okay. So for dihydrofolate reductase, there are some cephalosporins are there. Cephalosporins, or we can use the amoxicillin, which is the marketed drug, amoxiclav. So amoxicillin we use. So we have to see that if we give amoxicillin for to the bacterial dihydrofolate reductase inhibition, whether it is inhibiting the activity or it increasing the activity. That means whether it's showing the agonistic activity or antagonist activity. Okay, if the drug shows its activity and the bacterial enzyme get inhibited and decrease in the disease progression seen, then we go for the lead identification, sorry, lead discovery, or we can say that lead identification. So how we can find out the lead? So we prepare of set of 20 to 50 compounds. Okay, let me prepare a set of 20 compounds of different functional groups. Okay, different functional groups, but we have a core moiety. Okay, core moiety containing different functional groups. Okay, so now we perform the molecular docking. We perform the molecular docking. Then we find out various results. Some give minus 9.0, some gives minus 5.8, some gives minus 11.0. Okay. So when we find the when we perform the molecular docking after the preparation of the library, now what happens? We just go for after target validation. Okay, lead validation. Okay. So talking about lead validation, what in this step we do? There are 20 results we have. if we have a one setup library, we have 20 different results. From them, the highest binding affinity drug. From them, the highest binding affinity drug, let's suppose minus 11.0, is shown by the nitro. Okay, nitro group containing functional drug, and we have another drug is showing minus 10.5. During the validation of target, the amoxicillin we used or the cephalosporin derivatives we have used. Now we have performed the lead discovery process. We prepare the setup library, then we perform the molecular docking, and now we have to validate that result. Okay, we take the top candidate after the molecular docking, and they known as the heat. Okay, they are known as the heat or the lead. Okay, after that we go for the redocking. We go for the redocking. Redocking is nothing but docking of the ligand molecule at the active pocket and docking of the amoxicillin of that active pocket. Suppose this is our receptor. Okay, let me give you an example. This is our receptor, and this is the active side, and this is another side. Okay, this is another side. Let me give you an example. When we perform the molecular docking, prepare a grid box at this position. Okay, at this position, we get a result for amoxicillin minus 11. Okay, we get a result of minus 11. We perform the same molecular docking at this position of the nitro derivative and it will show the result of minus 10.5. Now, after as it is showing the similar results, very much similar results, we go for the synthesis. We go for the synthesis of that ligand molecule containing the nitro group. Before that, we have to see whether it is having the good ADME property, that is the pharmacokinetic parameter, that is the absorption, distribution, metabolism, and excretion. And you know what? The main thing that all these steps are performed virtually. By virtual screening, we perform this. We have various softwares like Protox, ADMET Lab. So there are various freeware softwares. We are going to learn about that. So this is the process. Okay. If qualifying these ADME characteristics and another, there is a Lipinski rule. If this follows all these things, then we go for the synthesis. After the synthesis process is done, then we test it in an animal, which is the clinical trial phase. This one is the phase 0. If animal trial gone successful, we go for the phase 1 to phase 3 study, which is the study on human beings, and then your drug is get approval by FDA and you got money of patent. Okay, you just patented, file a patent of that this molecule I have been discovered, and this is approved by FDA. Okay, so this is the whole journey of the of the drug computer aided drug design to human being. Okay, we just virtually created a molecule, then we perform all these things, we go through all these steps, then synthesis. There is also some challenges are there during synthesis. Might possible that compound is not getting synthesized easily, it cost more manpower, more chemicals. That is also one drawback. Okay, all things have their drawbacks and their some pros and some cons are there. Next, what is a target? So basically, target molecule is nothing. It may be protein, it may be enzymes. Okay, it may be enzymes. It's the receptor, the gene. So target molecule of various types are this. Okay, so it may be DNA also. The DNA may be the target. Okay, next. So how the target look like? So all of you know that the inside the cell we have chromosomes. From the chromosomes the DNA is prepared, DNA molecule. Okay. And there is another thing that is present inside your cell, that is the ribosomes. So ribosomes generally prepare the protein by the process of what? That's the translation process. Now, now these proteins are generally prepared of what? The amino acids. It is generally prepared of the amino acids. The amino acids combine to form the peptides. Okay, I just a small set of amino acid, a small set of amino acid is called as the peptide, and when the peptides combine each other, they prepare the protein. Now this protein, and you can see here, this is the receptor protein. Okay, and also they prepare the enzyme. Okay, enzymes are proteinous in nature. You have all learnt about it. Okay, by this process, the proteins are prepared from the amino acids, and there is also gene are there. There is DNA is there. Okay, so this is all about your target. Now, how to find out target? There are some sort of softwares are there where we can find out the target tools are there. The first one is the RCSP-PDB, the most commonly used software. It is called as the Protein Data Bank. It is called as Protein Data Bank. The PDB stands for Protein Data Bank. There is another Swiss Target Predictor, Drug Bank, CAST-P, Binding, Drug Binding. Okay. So these are some of the tools which are helpful for the finding of targets. We can go through each of the tools during this session. Okay. So now next talk about the target validation. As I've already told you, target validation is the process of proving scientifically modulating a biological target protein or enzyme or receptor will have a therapeutic effect. We just give the marketed drug, it will modulate the receptor. If we have a receptor, it will interact, the drug candidate will interact with the receptor, and there is change in the target takes place. If it inhibition takes place, then that is the plus point for us. Or if there is no activity, then the target is not completely validated. Why it is very much important? So because it ensures the target is release relevant. It reduces the last stage failure in clinical trial, because what we are doing, we are preventing the target prevention virtually. When it will pass, when it will pass the virtual process and we go for the clinical trial phase 1 to 3. Okay, if at that point this protein is not inhibited by the drug, so there is very much loss of the manpower, very much loss of the ligand molecule preparation chemicals that takes place. Okay, so it is also for the confirmation of the target is a safe to modulate. If we are modulating the amino acids and modulating the receptors, and there is some adverse effect is showing, so that is very much ineffective and very much costlier for a human being. We modulate the amino acids, but at the same time, this modulation can lead to any sort of other disease to develop. We stop the flu, but there is another disease develop inside the body by just modulating the receptor. That is also one type of problem. So we need to validate the target. Next, it is the common to save money and avoiding the wrong drug pathway. Strengthen the confidence before docking at CADD. So these are some of the points that are needed for the target validation. Now next, talking about the ligand preparation and optimization. I hope you are going to a great way. You are learning all these things and it will be very much enjoyable for you. So in ligand optimization, we have to first find out the lead, as I have already mentioned a lot of times. So these are the setup library. We have to prepare a library. Some sort of machines, automatic robotic machines are there, that is called as the high throughput screening. Then we'll discuss about what is chemoinformatics, library, drug likeness, what are the rules of drug likeness, what are the database to find out the ligand molecule or to prepare the ligand molecule. So what is lead discovery? So basically lead discovery is, it is the step where you find the potential starting molecule that shows the activity against a target. It is nothing but the to predicting the binding efficiency of the ligand molecule with the target. Okay, this is the lead discovery process. Secondly, now talking about the HTS, high throughput screening. So now I have given you here an example, the pyridine moiety containing the amino cyano group, and here it is the, we have prepared a set of 20 to 100 library. Let's suppose we have prepared 100 set of compound of this derivative, set of derivatives of this compound. Means what I am going to say, that this portion, this portion will remain the same, and we just change the R. Okay, we change the R with many such functional groups. Instead of preparing such function, instead of virtual screening, okay, we have our virtual screening process, molecular docking process, but HTS is not virtual screening. It is, we have to prepare such compounds, such derivatives here, and just by using some high advanced robotic technology, this HTS is done. Okay, by using some robotics mechanism, we just prepare many derivatives and we go for the go for the target lead identity validation process by using the HTS. We'll discuss in brief later also. So what are the features? HTS. HTS, high throughput screening and virtual screening or the molecular docking. What is the difference between this? It is a wet lab experimental screening process. So HTS nothing but it is a wet lab process. And the virtual screening in silico computational screening is where it is done by physically using the robots. Okay. So what is the input? The real chemical compound, we input the real chemical compound, the reaction takes place, and finally all these things are done by the robots. Okay. So output is the biological heat. As I have mentioned, heat is nothing but the most successful binding affinity results, more successful drugs. The cost is very high, so that is why the HTS process, we need robotic technology. First thing, we need robotic technology. We need very much amount of chemicals, prepare such huge quantity of derivatives. It is a fast process but limited by hardware. It is a robotic arms needed, plate readers are needed. The accuracy, high biological relevance. We are just predicting virtual string what? We are just predicting this may be the result. But in HTS, it is highly relevance, highly accurate result is there. The purpose is testing the compounds in real assay. Okay. So talking about virtual screening, it is a computer software tool. We have to prepare a set of library, predict the heat, and only computer is needed. If we are performing the advanced search, we need some more graphic card feature in our laptop. It is extremely fast, and by using some sort of softwares, we just prepare the molecular docking. It is a predictive, need validation, unlimited virtual library we can prepare, and it is for the prioritizing compound before the lab testing. Okay, so this is the difference between the HTS and the virtual screening. Now next, talking about the chemoinformatics. So what is chemoinformatics? So chemoinformatics name suggests, break it down. Chemical information. Chemoinformatics is nothing that is a information of chemical data sets. Chemical data. Okay. So we have to prepare a large set of database. So we have to remember all these derivatives. Okay. For that we need a software or any tools. Okay. So chemoinformatics helps to manage the large set of database to predict a molecular property. We have prepared one derivative, so we have to find all the properties. First we have to find out the pharmacokinetic properties, that is the ADME property. Then we also have to find out the toxicity profiling, how much toxicity is there, what are the toxicity, are the neurotoxic, hepatotoxic, nephrotoxic. So we have to find out all this toxicity property. Then we also have to find out the, there is another thing, is it following the drug likeliness, is it drug likeliness or not. So these are some of the properties we have to find out. We have to prepare a excel, as we prepare some excel sheet to maintain the records. So we have to prepare a database where we can record all the properties, then identify the drug likeliness molecule, perform the virtual screening, create a QSAR, that is the quantitative structural activity relationship. The term may sound very much hard to you, but QSAR is nothing but a then change in the position, change in substitution. So that is the QSAR. Design better ligand molecule, visualize the chemical structure in the 2D format or in the 3D format. Okay, so these are the things that are involved in your chemoinformatics. Next, what are the applications of chemoinformatics? The main focus of chemoinformatics to prepare the derivative of small molecules. Data, it is the storage of data of the chemical structures. What are the tools we use? Pyrex we use, OpenBabel is there, Autodoc tools are there. So the main use is drug design, QSAR. And there's another, that is the bioinformatics. So in bioinformatics, the main focus is on DNA, RNA sequencing, biomolecules. Then we just find out the sequence, change in the sequence, what are the structures. We use some advanced tools, that's a blast is there, uniport, clusteral is there. So these are some of the tools, like here as we are using Autodoc tools, Pyrex. In bioinformatics we use some sort of tools. What are the uses? Target identification, alignment of the target. Okay, so what are the chemical database we are using? I have also told you in that we have to study about database. So there are some sort of database, that is a PubChem, which is universally used PubChem. Then we have ChemBabel, Zinc database, ChemSpider. So from these four, this PubChem is very much important, and this Zinc database is also very much important for you. Chemical structure we can draw. We need also the smiles, the representation, linear representation of the compound, the smiles. Then we need the 2D or 3D structures of the chemicals. Okay, and the virtual screening, it is a structure based, ligand based screening. Okay, so these are some sort of things which we are needed for the ligand optimization or ligand preparation. Then coming to what is ligand molecule preparation. So ligand preparation, means converting the raw ligand molecule structure into a clean, chemically correct, energy minimized, suitable for the docking. So while preparing any lead molecule, if we have a structure, very easy structure, phenol. So this is just a 2D representation. To make it realistically 3D, I have to give it charge, which will be done by the tools I will told you. That is a chem sketch we can use, Autodoc tools we can use, ChemDraw we can use, and to make it completely valency, to satisfy all the valency. Okay, the carbon makes four bonds. The carbon that makes four bonds. If I just attach here one CH3 group, that is not good. That the valence is not satisfied. Okay, so we have to minimize the energy. We have to make it clean, chemically correct. The molecular formula, molecular structure should be correct. The energy minimization I will told you in the autodoc session, and suitable for the docking. What are the steps? So use the ligand molecule preparation. We use some sort of tools. We generate the 3D structures. Then we add the hydrogen, fix the valency, convert into the PDB format, protein database format for the Autodoc. So these are some sort of things or the steps which we are needed for the preparation of the ligand molecule. The next, how to prepare libraries. By using the same these softwares, we can prepare a setup library. We just call what are the process to prepare a library. If we need to prepare a library of known compounds, we just go through PubChem. Then we let's suppose we have a drug. We have to prepare some sort of drugs of the CNS activity drugs. Okay, we have alprazolam. We just type the alprazolam, then collect it smiles and paste it in the text notepad. Okay, we have to collect the smiles. By this process we can prepare a set of library. Also by converting the structures into the pdb format and save it, that is also a library. Now the main term here, the drug likeliness. There is a term is called as the drug likeliness. So what is this drug likeliness? So drug likeliness is nothing but whether the molecule have characteristics to become a drug. Not all the molecules can be drug. It should follow some specific points to become a drug. The first point is here. The molecular weight should be less than equal to 500 per mole. The number of rotatable bonds should be less than 10. Okay. Then the third one is the hydrogen bond donor should be less than 10, sorry, should be less than 5, and the hydrogen bond acceptor should be less than 10. So the log p value should be less than 5. Log p is the lipophilicity. The topological polar surface area should be less than 140, less than 140 angstrom. So these are the some sort of rules, 5 rules, which is very much important for a compound to be drug. Okay, that is a drug likeliness property. In case of CNS related drugs, we have some sort of drug which is very much effective toward the CNS. We are preparing some sort of drugs for the CNS. To cross the to get the brain, the CNS drug should cross the BBB. This stands for blood-brain barrier. it is highly lipophilic in nature. Lipophilic. So in that case, the log P we can we can make out that log P1. We can exclude that log P because for reaching the blood-brain barrier, for reaching the brain, the drug should cross the blood-brain barrier. So the log P may be lesser, it may be vary, it may be more than 5. So that is acceptable. Okay. So this is it for today's session. Thank you all for joining the session, and I hope that all of you learned what are the things which I have taught you today. The 3D representations and all the database representation will be done by in the next session, where we will study about RCSP PDB. We'll learn about zinc database, PubChem database, how to convert the structure into smiles, or how to convert the smiles into the structure. We will learn about the ADME property, that is the pharmacokinetic parameters. So this is all about for today's session. Thank you so much for joining, and see you soon in the next session. Thank you.